So we now are going to look at what we do with DNA. So as you know, DNA is the genetic information. So that's all the information that makes us and other organisms, that makes them what they are. So what happens with this DNA? We have this process called the central dogma. And the central dogma is just what they call the flow from going from DNA to the proteins. And the proteins are what is used to do everything that we need in our body. The proteins make all of our organs, makes all of the organelles, it's the enzymes that gets our body to work. So the central dogma is how do we get from DNA to those proteins? And there's mainly two steps. You start with DNA, you have to go through transcription to make mRNA, and then from mRNA you can make our protein. So we'll start with transcription. And one way to remember what happens in transcription is you think of that word script, which means to write. So we're going to write a message. So our message is mRNA, or messenger RNA. And if you look down at this diagram, here is our mRNA being built from the DNA strand. So transcription is a lot like DNA replication, that we have our strand of DNA that gets unzipped. So it has the same enzyme function happening as DNA replication. We have a helicase enzyme that comes in and unzips that double helix. And then DNA polymerase is the enzyme that adds the nucleotides to make the RNA strands. The major difference between DNA replication and transcription is that we're only making a short strand, a message. This message is just for one gene, for one trait. And DNA replication, we're making a whole copy of the DNA. This one's just a short strand. So there's another difference between, or a couple other differences between RNA and DNA. We'll look at that real quick. So the first thing is that RNA is a single strand, whereas DNA is our double strand. And the other main difference is in the nucleotides. RNA has 4 nucleotides, like DNA, which has 4 nucleotides. And they both have cytosine, they both have guanine, they both have adenine, but down here, RNA has uracil, and DNA has thymine. So now what happens at the end of transcription? At the end of transcription, we have our mRNA strand, and this is inside the nucleus. This occurred in the nucleus because that's where our DNA is located. So now the mRNA strand leaves the nucleus, and it will find its way to a ribosome. The ribosome is a location of protein synthesis or translation. So if we take a closer look, here's our mRNA strand, and we see our exposed nucleotides, and there's our bases. And remember that RNA has a U for uracil, so thymine. So translation occurs with the help of tRNA. So tRNA carries amino acids. So the tRNA has at the very bottom something called the anticodon, which will match up with the codons. The codon is 3 nucleotides that code for an amino acid. So the anticodon finds the codon on the RNA strand and brings the amino acid to the ribosomes. Those amino acids will join as they're being built with a peptide bond. And as they're being joined, they are creating this molecule called a polypeptide. And that is what's going to end up becoming the protein. And as you remember, we use these codon wheels, and that's how you can read the codon and figure out what it's coding for. So we have we always start with our first letter in the center and work our way out. So a codon of G, U, G would give us the amino acid valine. So once we have this polypeptide chain made, it's going to go through a process of folding to get our final protein. And this occurs in our endoplasmic reticulum. And inside that endoplasmic reticulum, there are these spaces, and that's where the folding occurs. So the polypeptides are made on the protein, or in the ribosomes that are on those endoplasmic reticulum. You can see the ribosomes there. They go into the space, and that's when the folding occurs. So that's how we go from DNA to protein.